All right. Well, thank you, everybody. The last day of February, and we're going to go out with some viruses. Pretty exciting. I'd like to introduce our seminar speaker today, Dr. Kim Thamatrakoln from Rutgers University. She has a BS from UCSD in biochemistry and cell biology, an MS from Stanford in biological sciences, and PhD. She did marine biology at UCSD, and she was there at Scripps. And she worked, Mike Latz was her, I think, advisor on paper. She really worked with Mark Hildebrand, who was the first person to isolate the silicon transporters that diatoms use to bring silicon into their cell. So she comes from a cool lineage in diatom research. She did a postdoc at Rutgers University, and then she remained there as research faculty for 11 years. So that's a long time. So for those of you that don't know, that means that she wrote grants and supported herself for 11 years at Rutgers, which is pretty amazing. Now she's been hired. She's ladder faculty at Rutgers. She's gone through assistant and she's associate professor now, much deserved. And we met a couple years ago now, 2012. And got lots of fun stories, but I think one of the ones that I enjoy most is she was here and she wanted some coffee, and I said, well, I have this mug, and then I have my mug, and then I have this mug with Seahawks on it. I'm like, she's like, I can't. And I'm like, are you serious? She's like, no, I'm not drinking out of that, because she's from San Francisco, which apparently, yeah. So I realized how deep football ties run, even for Kim, and we had never talked football before, but she would not drink out of Seahawks mug. So that is one of numerous stories I have with Kim. But the title of her talk is Shunt or Shuttle: Dynamic Biogeochemical Consequences of Diatom Host Virus Interactions. And I'd like you guys to give a hand to Kim.
Well, thank you very much. I really appreciate the invitation to come and speak to you. Thank you, Jeff, for inviting me. I'm especially grateful that this seminar is in person, because 2 years ago, I swore off my last Zoom seminar and I won't do it anymore. So I'm really happy that it's in person, because it's just much nicer to be able to interact and see people. So as Jeff said, this is the title of my talk, and there's a lot of jargon in there. And so I wanted to just spend the first couple of slides just making sure we're all on the same page. And I'm sure that you guys don't think about some of the things I'm going to talk about as much as I do, probably. So hopefully you all are familiar with phytoplankton and the role that they play in biogeochemical cycles in the ocean. And I'll try to remember to use my mouse here. So one of the things that we're interested in understanding is how phytoplankton mediate these biogeochemical cycles. And when we think about, and mostly with an eye on carbon cycling. And so there are three major pathways by which carbon can cycle through phytoplankton in the ocean. So we know that phytoplankton take up carbon dioxide, process it into their cell parts into organic matter. That organic matter can be transferred up to the higher trophic levels through grazing interactions, through fecal pellet production, and through just sinking and aggregation. Carbon can be exported into the deep ocean, where it can be sequestered. But then carbon can also return to the dissolved organic phase into the microbial loop and feed bacteria and archaea and other organisms, other heterotrophic organisms. So even if you don't think about phytoplankton as much as I do, I'm sure you're familiar with the idea that phytoplankton are incredibly diverse. So we've got two, they're represented by the prokaryotic cyanobacteria. So Prochlorococcus and Trichodesmium are the most probably famous ones of the prokaryotes. And then we have all the eukaryotic phytoplankton. So you have the green algal lineages, the chlorophytes, and then these red plastid derived lineages that encompass the dinoflagellates, haptophytes, and diatoms. And these are just some pretty pictures that show sort of the diversity. But when we think about carbon cycling in the ocean, carbon export in the ocean is predominantly mediated by the ballasted organisms. And so that's what's shown here in these SEM images. So as a whole group, the phytoplankton produce about 50% of the oxygen on the planet, but most of the carbon that's exported in the ocean is mediated by these three organisms. So dinoflagellates, haptophytes, mainly the coccolithophores that produce calcium carbonate shell, and then diatoms, which produce a silica shell. And that's because these mineralized phytoplankton produce cell walls that have ballast in them. And so they're heavier, they're denser than the density of seawater. And so they can sink to the bottom of the ocean. And so it's been estimated that the coccolithophores and diatoms together can account for about 98% of the carbon export in the ocean. And so this is sort of where this is my interest is mainly focused on diatoms. We do some work on coccolithophores, but today I'm going to just talk about the diatoms because I don't think Jeff would be happy with me if I talk about coccolithophores. So one of the things that I have a long-standing interest in that dates back to when I was a graduate student was understanding how diatoms mediate silicon cycling and silicification. So if any of you are familiar with Jeff, that tells you why we are connected, because he obviously has a long-standing interest in silicon cycling. So just a few sort of tidbits about the silicon cycle. So diatoms as a group among the phytoplankton, they're responsible for about 40% of the marine primary productivity, and they do form the link between the silicon and carbon cycle. So my early days working on diatoms I was really interested in understanding this production side. So what factors led to the production of silica, and mostly focused on the cellular and the molecular regulation of silicification. But then there's this whole dissolution side, so the recycling and the regeneration of dissolved silicon that can be utilized by other organisms. Silicon is limiting in most of the surface ocean, with the exception of probably the Southern Ocean. And there's a very important role that bacteria and zooplankton play in facilitating this dissolution process. And this dissolution, zooplankton contribute to dissolution because when copepods are sloppy eaters, and so when they eat diatoms, they sort of just leave some of the bits and parts, and that silica can dissolve. But work that was shown by a colleague of mine, Kay Beidel, as a graduate student with Farouk Azam, showed the importance of bacteria in mediating that dissolution. We're going to get back to that later on in the talk, so I'm just going to leave it at that for now. Dissolution is important. So silica recycling is critically important for maintaining diatom populations. It's estimated that dissolution or recycled silicon supports up to 80% of all diatom productivity. So you can kind of think of this similar to new and regenerated production that's normally driven by nitrate and nitrogen, ammonium, but a very kind of similar idea that it's the recycled silicon that can really be supporting a lot of diatom populations. And then this is just, so diatoms, because of these ballasted cell walls, these silica-based cell walls, they've been estimated to account for 0 to 100% of carbon export in a system. So that's a really cheeky way of just saying we have no real understanding of what constrains that. So they can either contribute all of the production, all of the export, or they can contribute some fraction of it. So one of the things that I started getting interested in, because I was interested in understanding sort of how diatoms mediate carbon and silicon cycling, I started getting interested a few years back in the role that viruses play in this process. So viruses that infect diatoms and induce lysis and mortality, how are they interacting and either short-circuiting or disrupting the silicon cycle through either processes by disrupting maybe silicification, processes that might mediate this dissolution or regeneration process, or how viruses might contribute to diatom-mediated carbon export. So viruses play a multifaceted and dynamic role in biogeochemical cycling. So this is a schematic from a fairly recent review that I just wanted to highlight. And so a lot of the sort of canonical view that viruses play in ecosystem dynamics has generally been attributed to a viral shunt. And so when we talk about the viral shunt, what we're saying is that phytoplankton or bacteria that are depicted here get infected by, so if they're not infected by a virus, their organic matter can be transferred up to higher trophic levels through grazing interactions. That's kind of shown here. But when viruses infect these host organisms and cause lysis, so they cause these cells to burst open, that material is shunted away from higher trophic levels and back into the microbial loop. So it's a way to recycle. So viruses have always been thought of as efficient recyclers. And this is fairly easy to kind of envision. So you have a picture like this. So this is some microscopy images. This is showing a bacteria cell here. So you see a single bacterium here, and it's being docked and infected by a virus. And after some period of time, you might be able to see some viruses being replicated on the inside. And then after a period of time, that host bursts open, it releases all these virus particles into the environment. And that's what we're referring to as the viral shunt. So you see all that dissolved organic rich matter that's being released and made available to the microbes. There's another potential route, another potential role that viruses can play in ecosystems, and that's one of being what we call the viral shuttle. And so this is when viruses infect a host and do something to the host that would end up facilitating syncing. And so this can happen through the viral viruses infecting a host, and like when we get sick with a cold, we get really snotty and mucusy and sticky. And so the same thing happens in phytoplankton. They get really sticky. They produce a lot of extracellular polysaccharides. And that causes them to aggregate. And when you aggregate, you can sink faster. So we would call that a process by which if viruses go in and infect a cell, cause this aggregation, that's a process by which we would say that viruses are mediating a carbon export event or a viral shuttle. And there's some other ones that we'll talk about later, that ways that viruses can sort of mediate that. So hopefully now all of that jargon that was in my title, you sort of have a vague familiarity with. We're talking about processes that facilitate the shunt or the shuttle, so carbon recycling in the ocean or carbon export. So how many of you have seen this picture before? Not that many? A few. There are your students. So this is a fairly, I thought, well-known image. So this is a drop of seawater. And to this drop of seawater, they added a fluorescent dye called cyber green, which binds to double-stranded DNA. And so this was a picture that was published by Jed Furman back in the late 1990s. And it was really the first time that we sort of could see the just sheer numbers of DNA-containing organisms in a drop of water. So you have a diatom here. Hopefully some of you recognize this pennate diatom. I'm sure Jeff did. All of these larger dots here are bacteria, and all of the smaller dots that are just way more abundant are viruses. And so this was one of the first times that we saw this, what they referred to as the microbial galaxy of viruses. So viruses outnumber bacteria in the environment by roughly an order of magnitude. So, and we can see that in an image like this. And so one of the things that makes diatom viruses, if you have studied diatom or viruses in biological oceanography or marine biology, I don't know if Jeff teaches viruses in his class. Do you? He probably talks about phage. We talk mostly about phage. So phage are viruses that infect bacteria. And that's probably what the vast majority of these are, phage. So because bacteria are so abundant, there's a lot of hosts around. And so what you've probably heard a lot about when people talk about viruses, they're talking largely about phage. But one thing that's really unique and interesting about diatom viruses is that they are among the smallest viruses on the planet. And so this is a series of scanning or transmission electron micrographs of different viruses. Most of these are marine viruses, with the exception of SARS-CoV-2. I put that up there just because it puts it into context for everybody. But the largest virus on the planet is this virus here. It's called the Pandora virus. It infects a freshwater amoeba. And so this actually rivals the size of a bacteria. And then we have viruses that infect coccolithophores. This is the Emmeliania huxleya viruses. Here are viruses that infect cyanophage. And then you have viruses here on the far end of the spectrum of viruses that infect diatoms. So this is about, just for context, this virus, I have my scale bar here. So this virus here is about 200 nanometers in diameter, and the diatom viruses are about 20 to 40 nanometers in diameter. So smallest on the planet. The other thing that's really unique about diatom viruses is that compared to the other algal viruses, so all of these other algal viruses, these viruses have genomes that are comprised of double-stranded DNA, just like you and me. But diatom viruses have genomes that are comprised either of single-stranded RNA or single-stranded DNA. So they're very unique from a genomic point of view. For those of you, you might be familiar with the virus that causes COVID. This is a single-stranded RNA virus. And so when we look at pictures like this, if you remember when I presented this, I was very deliberate to say this is a fluorescent dye that stains double-stranded DNA viruses. So you can't see diatom viruses on here at all. They're not detected by this stain because they have genomes that are not double-stranded DNA, but they're also too small to be resolved by epifluorescence microscopy. So if you do some back of the envelope calculations, we estimate that the average double-stranded DNA viral abundance in the ocean is about 10 to the 7 viruses per mil of seawater. If you take a typical diatom bloom that can be comprised maybe of 10 to the 4 cells per mil, blooms can get much, much higher than that. They can get up to like 10 to the 6 per mil in the environment. But let's just say a typical 10 to the 4 viruses or cells per mil in a bloom. We know from laboratory studies, we have some host virus systems in the lab, diatom host virus systems in the lab that we can cultivate. So from those systems, we know that during a viral infection experiment, a single cell can produce up to 10 to the 4 viruses per cell. So a cell gets infected, 10 to the 4 viruses can be released from that cell. If only 10% of a diatom population is infected, that would produce a viral concentration of 10 to the 7 per mil. So it means that this number of viruses that we estimate, we usually use this number of the number of viruses in the ocean is 10 to the 7, could actually be twice that many because we're missing all of the diatom viruses. And this is what makes diatom viruses really fascinating to me because they're completely enigmatic, undetectable up until recently. In the environment, we just know very little about them. And they infect one of the most ecologically important and impactful phytoplankton in the ocean. So some of the early work that I was interested in is like I said, I was interested in sort of factors that were affecting silica production and silicification. And so one of the things I wanted to do was I wanted to just ask, well, what are 2 conditions that we find in the ocean that we know impact silica production? And how would viral infection dynamics play out under those 2 scenarios? So this is a global map from this study back in 2016 that shows that attempts to highlight what the most limiting nutrient is for diatoms in the global ocean. And what you'll see from this picture is that vast majority of the ocean is limited by iron. So we know phytoplankton need for photosynthesis, and so they're very greatly limited by iron. But they're also limited, not surprisingly, by the availability of dissolved silicon, which is depicted here as silicic acid. So diatoms experience widespread silicon limitation, and we know that when diatoms are silicon limited, they can decrease silica production. So they can actually lower the cell quota, the cellular quota for silica, and they produce less silica. When diatoms are limited for iron, it's been shown in some species that can actually increase silica production. And so we have these 2 contrasting scenarios of silicon limitation and iron limitation that have differing impacts on silica production. And we just wanted to know, well, what happens to viral infection under these conditions? So like I said, we have a couple of different diatom host virus systems in the lab. This is just one of the ones that we have. It's a diatom called Catastris tenuissimus that's shown up here in images. This particular Catastris species is a solitary cell. They can form these really long, beautiful chains. And we have 2 different viruses that infect this host. There's a virus that we call C10 DNAV, which is a single-stranded DNA virus, and then C10 RNAV, which is a single-stranded RNA virus. And so we set out to do a pretty simple experiment. We took cultures of Catastris tenuissimus. We grew them under iron limitation or silicon limitation. We infected them, and then we looked to see what happened to the dynamics. So what we found was this. So this is just a plot of host abundance versus time. We grow the cells for a period of time under these conditions, and then we add viruses. In this experiment, we added viruses here by this arrow. So these solid symbols are the uninfected controls. The open circles are the infected samples. And then we have the replete, so our normal control media shown in blue, the silicon limited samples in green, and the iron limited samples in this orange. And what we saw was that under silicon limitation, viral infection and lysis happened. It's hard to see on this because they don't grow. Obviously, they need silicon. But the lysis happens faster under silicon limitation, and it happens slower under iron limitation. And I'm not going to actually spend a lot of time on these. These 2 studies are published in these 2 papers. So if this is an interesting story to you. I encourage you to check those out. But it allowed us to develop this working hypothesis about how the shunt and the shuttle might be operating under these two contrasting nutrient scenarios. So what we were sort of proposing is that viral infection under silicon limitation, because it happens faster and the mortality is more severe, that would tip the balance towards the viral shunt and recycling in the upper ocean. But under ion limitation, because the viral infection is impaired, mortality is reduced, and we combine that with some observations in other studies that show that iron limitation can increase silicon production, which makes the cells heavier. We propose, or we posited, that under iron limitation, viral infection would actually facilitate the viral shuttle. What time? I don't have a. So this is what we've sort of been working on, is trying to understand this relative balance between the viral shunt and the viral shuttle. And so I started thinking about sort of processes that were specific to diatoms that would either facilitate the shuttle sinking or the shunt. And so I'm going to start by just talking about some of the work we've done looking at processes that we've been looking at that would facilitate the shuttle. There's probably more, but the ones that we were particularly interested in was the role that viruses play in increasing silica production on a cellular level, whether they induce spore formation. So diatoms, part of the diatom life history is that they can form into spores. These are resting spores, and they do this when conditions are not ideal. And what it does is it forces them into a resting stage. They're very low metabolic activity. They get very, very silicified. So they become very heavy. They sink into the sediments. And then through upwelling events, when times are more ideal, they can be brought back into the surface where they can resume, they become vegetative and they can resume growth. So that was one thing we looked at. And then whether there's a role of aggregation that's mediated by extracellular polymer production or aggregation. So these are some of the things we've been looking at. We did a study in collaboration with Marina Montresor at the Stazione Zoologica in Naples, Italy, with her then graduate student Angela Pelusi, who was studying spore formation in diatoms. So this is a different diatom. This is Catastris socialis, which is a chain-forming diatom. So you can see this is a 5 individual chain, and they can produce these spores. This is what a spore looks like of Catastris socialis. So this is a very heavily silicified resting spore. And what Angela found was that when she added viruses to these cultures, she saw that there was, you can just look at these bars here. So this is the percent of the culture that transitioned into spores during the infection. And so what she found was that up to 50 to 60% of the culture went from a vegetative state into a resting state. And then what she did is she took those resting spores that presumably had viruses in them, and she returned them under ideal conditions. So she removed the viruses from them, she put them into fresh media, and she looked to see whether they would regerminate. And she saw that they did indeed grow. They resumed growth, as you would expect, but no viruses were actually produced. And so we sort of proposed this as a potential defense mechanism that this particular species might have, that when it gets infected, it can actually transition into a resting spore phase, which actually ends up truncating the viral infection cycle and doesn't produce any viruses. So this study has been published. So if you're interested in that, I encourage you to check out that publication. Some unpublished work that we have some preliminary observations that diatoms that are infected have a higher silicon to carbon ratio in their cells. And so if we just look at sort of these box points here, we see the control cultures in blue and the infected samples in orange. And we see that the silicon to carbon ratio in the infected cultures are about 2 to 3 times higher than silicon to carbon ratio in the control cells. And so this is another mechanism by which viral infection may facilitate sinking, because you're increasing the amount of silicon to carbon. We also had a student who was interested in looking at the role of aggregation mediated by TEP, which is transparent exopolymers. So these are acidic polysaccharides that are very, very sticky. We can stain TEP. The TEP is transparent. So we can stain it with a stain called alcium blue, which makes them blue. And what you're looking at here is a particle that was produced during an infection. And you can see all these cells that are sort of occluded within that particle. And so what she found in this was that infected cultures made more and larger particles. So you get this aggregation event. When you get aggregates that are ballasted with diatoms, these aggregates can actually sink very, very fast. So that's some of the work that we've looked at in terms of the viral shuttle. But I want to talk about some of the work we've been looking at to look at the viral shunt. And so when we think about the viral shunt, as I said earlier, we think about the infection shunting that energy away from higher trophic levels and back into the microbial loop. And one of the reasons why this is really critical is because I mentioned on that first slide that dissolution processes can be mediated by marine bacteria. And so this is, again, work that was done by Kay Beidel and Farouk Azam. And what you're looking at here are some really pretty epifluorescence microscope images. And so this is a diatom, I think it's Cylindrotheca fusiformis, a diatom that's being colonized by marine bacteria here. So diatoms have this silicified cell wall that we call frustral. And in order to protect the cell wall from dissolution in seawater, they encase themselves in this organic matrix. So it's an organic matrix that's made up of proteins and carbohydrates. I mean, it just sort of buffers the cell from the extracellular environment. What bacteria do is they release these proteases and other types of ectoenzymes that can actually break down that organic layer. That's what they use for food. And in breaking down that organic layer, they expose the silica to the seawater, which facilitates the dissolution process. And so what you see is these diatoms that you can see the whole cells here. They've been colonized by the bacteria, which has facilitated that dissolution process. And bacteria can accelerate dissolution. Silica dissolution in the absence of bacteria would take months, maybe months on hand. But bacteria can accelerate that to being within the order of days or weeks. And this is just showing you some data from that. Should have put that up anyway before. But this is the proteolytic activity that's measured across different bacterial isolates that have been fed diatom detritus. And we see that there's a range. Not all bacteria will consume the organic matrix of a diatom cell, but you see that we can get very high proteolytic rates from some of these different bacteria. And if you look at the proteolytic rates versus the particulate silica dissolution, you see as the proteolytic activity goes up, the silica dissolution increases. And so this was sort of evidence that suggested that bacteria are sort of denuding that cell of that protective organic layer. And that's just a little schematic that I drew to kind of visualize what's happening. So bacteria are colonizing this organic layer, releasing these proteases, and allowing that silica to be dissolved by the extracellular. So this was the importance. So the reason why we started getting into sort of looking at dissolution and bacterial activity was because I wanted, like I said, I wanted to understand sort of how viruses were interfacing with the silicon cycle. And this was one way to look at how they might be facilitating shunt processes. But what we had observed on in an earlier study, so this is a project that we did with Jeff on a cruise in 2013. And so this is from this paper. So I'm not going to spend too much time on it. But what I want to sort of point to you is that what we were able to do in this study was we were able to determine that along a continuum of silicon stress, which is shown here on this x-axis, going from replete on the right to limited on the left, we can actually sort of trace the degree of infection along, we can sort of recapitulate infection dynamics along this continuum of silicon limitation. And so what this is basically showing is that, think of this in reverse. This is one of those examples where you publish something and you go back and you say, why did I publish it that way? I should have flipped this, but we did it in reverse. So you kind of have to start from the right, where you're looking at sort of an early infected cell. And as the infection becomes more rampant and there's more active replication of the viruses, the population reaches a peak biomass and a peak degree of infection. At some point, there's lysis that happens. The cells burst open, and all those viruses get released into the extracellular space, and the biomass declines. But the point that I want to make with this slide is that we measured proteolytic activity in these samples, and what we saw is that these populations, so this is shown in this this color plot. So these populations here that we diagnosed and defined as being post-lytic, so they had already undergone lysis, these populations were characterized by having some of the highest proteolytic activities. And so this led us to the hypothesis that viral infection stimulates bacterial-mediated proteolytic activity, which would enhance the viral shunt and facilitate silica recycling. So we wanted to, that was sort of the hypothesis that came out of this study. So we wanted to test this hypothesis in lab cultures. And so we set up this fairly simple experiment. And this is a call out to all the students in the group. Always start with a simple experiment because it's going to get complicated. And this certainly got complicated, but always start off simple. So what we did is we took cultures. So we took uninfected cultures and infected cultures, and through time, so we let the infection experiment run out and run its course. And in the infected cultures, what I'm depicting here are dead cells, and then these are the DOM. This is like particulate organic matter in the diatoms here. And then this is just kind of depicting various types of dissolved organic matter that's produced by the cells and released during infection. So upon lysis or upon death, this dissolved organic material becomes available into the extracellular space. So we collect the dissolved fraction, and we take this dissolved fraction in the infected cultures that includes the viruses, and we feed that to bacteria. So we feed the DOM to bacteria, and then we just measure whether those bacteria have higher proteolytic rates or whether they're able to actually use that material and use that for growth. So when we think about microbial production, secondary production, usually we're thinking about growth because we're thinking about respiration. But the other aspect is bacteria can only use organic matter that's of a particular molecular weight. So if the organic matter is too big, they have to release these enzymes to chop it up into smaller bits that they can actually transport. And so if the material is more labile, meaning more accessible, then bacteria would be able to grow immediately. But if it's less labile and more recalcitrant, they would need to process that in order to be able to actually utilize it. So we set up these experiments. So this is just showing you what the host dynamics look like during this infection experiment. So this is showing you in blue is the control uninfected cultures. And I said earlier that we have two different viruses that infect this host, a DNA virus and an RNA virus. And that's shown here. They have slightly different infection dynamics. The RNA virus takes a little bit longer to infect its host than the DNA virus, but they both lead to host lysis. So when we started this, we had our hypothesis that infection was going to stimulate bacterial like proteolytic activity. And so we did this experiment where we measured the proteolytic activity using a protease assay, which I can talk about if people are interested. And what we saw was really quite striking. We saw that the material, the dissolved organic matter that had been derived from an RNA viral infection stimulated proteolytic activity more than the dissolved organic matter that was derived from either control uninfected cells or from cells that had been infected with the DNA virus. So, and the colors here are just showing you the time points along the time course of the infection. And so as the infection progresses, we see that there's much more stimulation of that organic matter. So that kind of confused us because I thought, well, there's all this organic matter, the DNA viruses, you can see here that the DNA viruses are lysing the cells and presumably releasing organic matter. So why aren't the bacteria responding to that organic matter the same way? So then what we did is we looked at the specific growth rate of the bacteria in response to this material. And then we saw something that was even more confusing because we saw that the bacteria were growing when they were fed DOM produced from the control uninfected cultures and DOM from the DNA infected cultures, but they weren't growing from the material that had been released from RNA viral infection. And so we scratched, so I am not a microbiologist who studies bacteria. So this was my first foray, and I know a lot of people think that microbiology is all the same as long as they're microscopic, but I didn't have a really great understanding of bacteria. I don't work with bacteria, and I was like, I don't know what's going on here. This is like the experiment failed. We were totally showing contradictory data, and I don't understand what's happening. But what we did, so we kind of had this, at the same time, we have a close collaboration with Ben Van Mooy and his group out at Woods Hole. So Ben is an organic geochemist who's really interested in understanding lipid production and lipid cycling. And so one of his graduate students was doing some characterization of the dissolved organic matter of these cultures. And one of the things, so this is one of the things that we found, we were trying to understand what was going on. And so what's going on with the DOM? Is the DOM different? Is it compositionally different? Oh, so what I should mention, let me go back to this. What I'm showing you here is data from a isolated bacteria, so a single cultured bacteria. We actually showed this in 2 different cultures of bacteria, and we also showed that the response is robust with a mixed bacteria assemblage. So we did a lot of work. This paper is still in review, hoping to have it accepted any day now. But this is a really robust finding. So, because when we first saw it, it was contradictory. I said, let's try a different system. Let's try a natural community. And it was always the same result. And I thought, okay, well, this has to be real then, because it's really robust. So at the time we were doing this, we were looking at the dissolved lipidome. So looking at polar lipids that are produced during infection. And I show this mass spec. This is an HPLC mass spec data. And what we're overlaying here is basically the signature of control cells, DOM produced from control uninfected cells in green, the RNA virus infection in blue, and the DNA virus infection in red. And the point that I'm trying to make here is there is a different profile that's produced by these different conditions. So the organic matter, the landscape of the organic matter is different between cells, control cells. We also tested cells that just died due to nutrient limitation. So wanting to see if this was related to just death processes in general. And it's not, it's something very specific to viral infection mediated death. And so you see these peaks here of molecules that are produced only by the RNA viral infection, or only by the DNA viral infection, or only in healthy control cells. And we can take a little closer look at this. So this was a study that was published by a former graduate student of Ben Van Mooy, so Bethany Edwards, who's an assistant professor at UC Berkeley. And Bethany was really interested in understanding the oxylipin profile of the organic matter. So oxylipins are polyunsaturated aldehydes that have been implicated in a lot of cell-to-cell communication. So she had a different interest in it. But the point here is to show you this is a subset of oxylipins that she was particularly interested in. And this is the expression of those oxylipins during, I think it's DNA viral infection in this first column, and then RNA viral infection in the second column. And again, just to highlight that there is a different landscape of organic of dissolved organic molecules that are produced during these 2 infections. So how much time is it? Okay, perfect. So this led us to this conceptual model that we came up with to try to understand what the impact of these 2 conditions are on microbial processing and remineralization length scales. So I'll take you through this. So what we're showing here is this, on the left is the DNA viruses, on the right on the RNA viruses. So relative to DNA viral infection, what we're proposing is that enhanced proteolytic activity by RNA viral infection would increase the regeneration of nitrogen. So remember, we are releasing proteases, which are breaking up proteins, and releasing amino acids, which are really rich in nitrogen and are being used as a source to support primary production. This leads to a decrease in the F ratio. So this is the ratio of production that's supported by new nitrate versus regenerated nitrogen. It would increase silica dissolution, because as I showed you from work that Kay Beidel did, when we increase proteolytic activity, we increase silica dissolution. So this would increase silica dissolution and then increase the D to P ratio, which is the ratio of dissolved silicon relative to, that's the ratio of the amount of silica that dissolved silicon that's supporting production. And so you get a higher D to P ratio, so more silicon cycling. And then this would, by definition, because of this increase in proteolytic activity, would decrease the remineralization length scale. So the remineralization length scale is the depth at which organic matter is remineralized and can be reused by heterotrophic bacteria. And so if you decrease the relative, the remineralization length scale, which is shown by this sort of arrow in the background, the relationship of that remineralization depth relative to the mixed layer is what's going to determine whether that carbon is exported or maintained in the upper ocean. So if the remineralization length scale is more shallow than the mixed layer, that material is just going to cycle and recycle up in the upper ocean. But if it's deeper than the mixed layer, so the remineralization length scale is deeper, that carbon has the likelihood that it could be exported and transported to depth. And so this is sort of our working hypothesis. And I was supposed to insert another slide and I forgot to do it. So I started off my talk with a title that said shunt or shuttle. And my final slide said shunt or shuttle. And the answer is yes. So both of them happen. It's the environment that sort of decides whether material is going to be shunted or shuttled. But then this study has sort of added an additional layer to the shunt because it's not so straightforward as do you get upper ocean recycling? It actually may be a lot more complicated and dynamic than we think.